Recording. Alright, so this is the second session. Before we dive into the data, just some practical things. So next session is for next week. Same time slots as it is today. Then the week after that, RosBREED has its participant meeting, so I won't be able to give a session. And the week after that, I'm traveling, and it's really hard for me to give a session right that day. So the next session after that, session 4, will be on April 10. At that point, Europe will have changed to daylight savings time, so then everything is moved back an hour again for the people in Europe. Then I asked you to download some backup files. I didn't go over those last time. We only really start needing them this session, or hopefully not. But sometimes when I create input files, or you may create input files, some small errors may occur. You might have forgotten a space somewhere, stuff like that. Sometimes it's really hard to track a small little issue down, and it might take a little bit of time. If it's a bigger issue and a lot of people have an issue, please say something so then we can figure out together what's going wrong. But sometimes I've noticed it's very user specific, and it might be one person that has an issue. So at that point, I would like to ask that you, after the session, check what went wrong, and the backup files, which like I shared here for this session, have several input files, output files that can be used to quickly jump to the point where everybody else is at, and so that you can still follow. So for instance, under B allele frequency, it has a CSV file that will be used as input file. It also has the output file from the R script. So at that point, you can anybody who has an issue can quickly follow along with these files that have been made up front, and that should work perfectly. It sometimes also might be an issue with your software, and again, I'm more than willing to help figure out what it is, but it might not be best thing if we do it during the sessions, because that maybe everybody's waiting for one issue. And then one more thing that I wanted to say is that I've gotten the question from several people about the recordings of this. I'm more than willing to share it, but right now I'm looking into a good platform to do so. Ideally, it would not just be the file, it would be like online something where you can watch the video or the recordings, but that would then also have some information on the paper that describes this workflow that we're doing, and that would allow us to keep track of how many people are watching the video. So if anybody has a good suggestion on where I can post the recordings, please let me know through email, and I will look into it. So then we'll get starting with the actual session. So for today, we'll start doing our first curation steps, which means we'll look at individuals and their quality and how well they performed on the array, and looking at markers. So in this case, SNPs, and check which SNPs are working and which ones are not. And so as a reminder, this is the whole workflow. Last session, we talked about our input data and why it's important to go through this workflow. And so today we've gone at the start of the workflow, which will be the looking at the quality and looking at marker quality too, and sample quality. So why do we look at sample quality? Well, garbage in, garbage out. So if you have bad samples, things that didn't work well on the array, then probably your results are not so good, and whatever you do afterwards with them is also not going to be really reliable. There are several reasons why a sample might not work properly. Sometimes there might be, for instance, DNA concentration. The arrays or the facility we work with requires a minimum of 50 nanograms per microliter, and I think that's more or less the standard for the SNP arrays of Illumina. Your DNA might have been degraded, it might not have been really pure, it might not be your DNA quality, maybe you had some contamination and there's actually 2 samples in a single well, or we've had the case where one array just failed, the running of the array, so it wasn't up to the sample. It wasn't anything to do with the sample. It was the array and the chemicals or the chemistry of it that failed. So there are several reasons why, and it's why do we want to have them removed as quickly as possible? Well, if something went wrong with the sample and they're behaving strangely, then there's a good chance that might influence calls of other individuals, especially if you have several individuals that did not have good quality. And so by influencing the other individuals, actually you're kind of contaminating the other good individuals, as well as you might look at some markers and they may not look so good, but actually they are good. It's just there's some samples that didn't work well. And so by removing them at the very beginning, you avoid influencing all your other analysis. So how can we find individuals that have poor quality? And I'll open Genome Studio for that. So if everybody can open Genome Studio. If you open Genome Studio and you created a new project, or if you have created a project before, you get to this screen, and then to open your project, you just double click on the project you want to open, and then it should open in Genome Studio. So everybody should have a project by now that has all the data in it that I've been sharing, and so that would mean you have 77 individuals here if you look at the samples table. And to look at the first thing we can do to look at sample quality is to check how many of the SNPs gave actually a genotype call for an individual. So for every SNP, an individual gets called either AA, AB, or BB, or it doesn't get a call because Genome Studio figured it couldn't determine the genotype accurately. And so the proportion overall SNPs of how many types it got a call, so it got AA, AB, or BB, that's the call rate, and that should be as one of the columns here. And as you'll see here, for most of you, that will all be 0, which is not true, because if you look at these graphs here, you see that many individuals are called, in most cases all. So to get the call rate, you'll have to right-click anywhere in the SNP table and say recalculate statistics for all samples. And if you do that, that should go relatively quickly. Then right now everything still looks 0, but if once it's done, it will show the call rates. So here we go. There's a bunch of call rates. And so we can have a look at that. And now if you look at that, we see that many of them are around 0.8, but we already see one that is around 0.65. And if I want to know which individual that is, that's a bit far. So I'm going to grab this column by clicking on its name, and then I can start moving this around. And as you saw, the table jumped immediately to the beginning, and so I can put this somewhere here right next to it, so I can have a look which individuals have what call rate. And if I want to sort this, then I can press here on the A to Z button, which is sorting, ascending, so we'll get the smallest one on the top and then the higher ones at the bottom. And then we can see that there's a few individuals here that have a lower call rate than, or have a call rate in around 0.6 and 0.65, and then after a while this gets up in the 80% for call rate. So what would be a good cutoff value? Well, if you want to see how your samples overall behave, you can, in the sample table here, you can go to the histogram plot, and if we click on that, a histogram plot will appear, and because I already highlighted my call rate column, it's immediately showing me the histogram for the call rates. So, and if we look at that, we can see that almost all of our individuals are in this area here. There's a few that have a bit lower, and then there's some that have a much lower call rate. So if you look at this, I would say at least all these, everything up to 0.66, and probably even that one sample that has a value of 0.7, that is definitely lower than most of the samples. So I'd say there might be something wrong with this. Not necessarily though. You need to also know your germplasm. Why am I saying that? For instance, in apple, we've run some wild apples, while the array or the SNPs were identified using apples that were representative of the breeding germplasm. And so wild apples might have a lot of alleles that those breeding germplasm doesn't have, or their genome might be completely different, and so they might not always be binding very well to the probes of the array, and if they don't bind very well to the probes, they don't get a lot of amplification, and then for a lot of SNPs you might get no calls. That doesn't mean your sample was bad, it just means it might not have been really the target kind of individual for your array. So if I would find a wild cherry here in this list that has low call rate, then I would not be so worried about that sample. But given that these are like Napoleon or Rainier or Bing, those are pretty common varieties, they shouldn't have such a low call rate. So for these ones, I am a bit worried that they might not have worked that well. But there's other ways to look at this too, and so you'll gather evidence for whether a sample is working or not. So one other thing we can do, and let me just make Genome Studio large, is we can look at the SNP graphs here. And if I look at this graph here, I already noticed that there are some individuals that are far away from the main clusters, like these here or these here. And if I go through this, I see that that is regularly the case. So I can have a look at what those individuals are. So I can select them, and then either I can drag a box around them, like this, which is normally, you don't really have to click anything in Genome Studio to get the option to select with a box. You just click on your mouse and you drag, just like you would do in any other Windows environment. You can also use the lasso mode here on the SNP graph, and so you can then cover an area the way you want it around the SNPs that you want, and they'll all get highlighted. And as you can see, they all get highlighted as well here in the SNP table, so you can see which individuals you're selecting or choosing. And then as we go through, and maybe this one too, so if you want to get a second set of individuals added to your selection, you can hold Control, and then you select them, and they should be added to your selection as long as you hold Control. And so if you can go through more of the SNP graphs, you can see that yes, these individuals I selected might not always be so clear, but the yellow ones are often outside the box. And there's actually some more, so I can select a few more of these and just see if these, if they often are highlighted or not. And so as you can see here in the table below, I've selected mostly or all individuals with a low, or most of the individuals with a low call rate right now. And if I would go through more graphs and I would select some more individuals, at the end they would all be selected. Now, once in a while you see that here is one is chosen that has a higher call rate. So once in a while you might actually still have a sample that has a decent call rate. So the other way to get an idea how often they are outside of the clusters would be to look at it SNP by SNP. So in this case, I'll just choose this SNP. I'm not holding Control right now because I just want to have only the SNPs that are outside of the clusters in this SNP graph. So now I selected all of those, and I can get the names of these individuals into Excel by highlighting the sample ID column by clicking on its name here. once. If I click again, I unselect it. And then I click on the copy button, which is these two sheets here. And then if I open an Excel file, and I, do that. Okay. And I paste by clicking Ctrl V in Excel, then I get all these individuals. Now I can do that again for a different SNP. Let's pick this SNP, and I can select everything that is outside the main clusters, and I hit copy again here on these double sheets in the sample table, and I paste again in Excel. And if I do that a lot of times, I'll get an idea of which ones are selected over multiple SNPs. You don't have to go through all of them. You could do 10 or so, or 15. And if you then, I'll copy this whole column again to a new column. So I have them now twice. And then under the data sheet in Excel, I choose, let me widen it up. There should be an option to remove duplicates, and the symbol might look slightly different in your Excel file. But if I do that, it will remove all the duplicates, and so it will say I found 9, there's 11 unique values still here. And at that point I can see, well, of all the individuals I've been selecting over multiple graphs, how often do they occur? And I can do that by, on the cell next to it, I can use the formula count if, and as the range I choose the original one where I didn't remove any duplicates. So I make sure I have the whole column, and then comma, and then I choose the criteria, which is in this case this individual. I want to know how often this individual occurs in this whole, in the other column, and then it tells me that sweetheart is occurring twice. And if I look at this, there's some individuals that are occurring twice and some individuals that are occurring only once. Well, 2 is not really a good sample size, so you would need a bit more, but in that case you might see that an individual, maybe this individual only would have been chosen only once over the course of many SNPs, so that might actually still be a good, but then others might have been selected almost every time or all the time. And so that would be another way of looking at seeing that, yes, those samples are often far away from the cluster plot, so something might be going wrong with them. And then one more way, and the way that I like a lot, is by going to the full data table of Genome Studio, and I'll make this large again, and choosing the histogram plot here. So it's very similar as the histogram plot in under the samples table, but in this case it will allow us to plot parameters or from the full data table. So by clicking here, it calls the Genome Studio histogram, and then I want to go look at the B allele frequency. Now at this point the B allele frequency is grayed out, and that is because it is not how should I say, that is a property of a sample. So you have to first choose a sample in the main columns. So, and a sample can be recognized by it because it has a number behind it. And if you know your germplasm, you probably know the names of your samples. So by clicking on one of those, you see that this subcolumns become available. And then I click on the B allele frequency. Now, what is the B allele frequency? It's it's the frequency of how often it sees a B allele, or how much of a B allele it has seen for each SNP. And so this is then the histogram for all the SNPs. So it's very similar to the theta. And if you remember from last session, that is the x-axis, and that was saying the proportion of how much a B allele intensity was observed. but you see that in that case, we expect that most of them are either on the left side or on the right side or right in the middle, because in the left side it would mean 0, which means AA. On the right, it's all B allele intensity seen, so that means BB, and in the middle it would be 50-50. But you see there's a decent amount here in the middle, somewhere in between, and that's because, as I shown you last time, sometimes clusters shift around, and to my knowledge, the B allele frequency takes into account that shifting of the cluster, so you get kind of clearer peaks where all AA individuals are now really on the left and all BB individuals are now really on the right. Now, I've been going here between theta and B allele frequency. Now, if I would press the up or down button on my keyboard, you see that I'm just scrolling through the different parameters for this sample named Giant. But I don't want to go through all the different parameters for Giant. I want to go over the individual. So to do that, I just click on Giant again, so that my selection is in this window. And then if I press down, I can now scroll through all individuals. And as you see so far, we see nice 3 peaks. That all looks good. And then as we go down a bit, we hit Napoleon. And Napoleon doesn't have these nice 3 peaks. Rather, it has some blurry big thing in the middle. So that's not what should be happening. So this is really a clear indication that something went wrong with your sample. And we can scroll a bit more and we see some other ones, for instance Van, and as you can see, those are all the ones with the low call rate as well. But this is a really easy way to quickly scroll through your samples and to see what is happening. This is a slightly different one, but you can also see that there's not really a peak, rather there's just a flat stretch here. That's also not really good. And that also indicates that something went wrong with your sample. Now I'm gonna quickly go back to the PowerPoint because, and that's the wrong one, sorry about that. Here, let me close this one and open up this one, because there's a few things I cannot show right now with Cherry, and because I don't have examples of it in this data set. But here are very similar plots made for Apple, and you can see that we got more or less the 3 peaks, but then there's these small little peaks that arise from the side, and those might indicate that your sample got contaminated, so that there's actually DNA of a second sample in there. And I expect, I haven't really seen it, but I expect the more contamination, the more these peaks will shift towards this center. And why do these peaks occur? Well, your sample is truly BB, but if the sample that caused the contamination is AA, it's slightly off-putting that BB, and the same on the other side. And so, as the proportion, or as the amount of contamination changes, that will lead to a larger proportion or a larger deviation, because there's more of the wrong alleles that are binding. So that's one other thing that you may encounter, is these little shoulder peaks, and that could indicate contamination of your sample. The other thing, which I hope you don't have to see, is what I saw here with the Apple array. I'd never seen this before, but turns out we got a lot of samples from the USDA Geneva collection, and they have a big apple germplasm collection, and we requested a lot of apple samples from them, and we put them on the array. But we had also requested some cherry samples from the USDA collection, and most of those are in Davis, but apparently there were 3 samples that came from Geneva, and I think I'm not sure if they got shipped together, but in the lab nobody realized that those 3 samples coming from Geneva were not apple samples but cherry samples, so they got run on the apple array, and so that's what they gave us. So if you see a really weird pattern, something is wrong and you need to go and investigate what's happening. And in most cases it will just be something went wrong with your sample. In our case, it turned out we had some cherry samples on the apple array. Now I don't know if we do the opposite. We run some samples on the cherry array, if I get the same plots, or maybe I get something completely different. There is one more graph you can look at in the histogram plot, and that's the R rather than the B allele frequency. And so the R is the y-axis in your SNP graph, which is the total intensity observed. And for most samples it will look something like this, kind of a normal, not necessarily completely normal distribution, somewhere around 1.2, and then here a few, and then some around 0, and that's just SNPs that failed overall because they don't lead to amplification. But if you look at a sample that's not so good, you'll see that all of this is squished together, and that there's a series of SNPs for which the intensity is much higher. So that would be another way to look at your sample quality. And so then combining all that information that you got, the call rate and the B allele frequency and their position in the plot, you can start making a decision on whether you trust the sample or not. And you may say, well, a call rate of 0.6, that still means that there's plenty of SNPs that got called, maybe not as much as the ones that have 0.8, but I can tell you that even that 60 or 65% that is called, there's a lot of errors in there. There's a lot of things that are not right. We had a few of those samples in Apple, and when comparing them to their offspring, they just gave so many errors that I had to make them missing. Luckily, we had a lot of offspring, so based on the offspring, we could determine what their genotype was, and then later we reran them again, and then our imputations showed that those imputations were correct given the actual sample, and so that the sample with the low call rate that I had indicated as being a bad sample had indeed a lot of miscalled genotypes. So don't trust. A lot of these you can't even trust. So it's really the best way would be to remove them from your data set. The other way you can look at it is that I'm not sure what it costs right now to rerun a sample, but if somebody spends hours and hours and hours on it, fixing it, that might cost way more than rerunning the sample. And if I'm not sure where it is, it should be here. If you look at, there is a column now. If you use the sample sheet that I shared and asked to create a new project with, there was a column in there showing poor DNA, and that was based. It's not really poor DNA. In most cases, it was the array that failed, but it indicated that these samples are not good and that we should remove them. So if you want to remove them from your analysis, you can highlight them, and you just click on the first sample here, and by holding Shift and clicking on the last one, you select everything, and then you right click and you can say exclude selected samples. And so if you do that, it will ask you if you wish to update some errors, and at this point I'd say no, because the first thing, and it asks also for the SNP statistics, so again no, because the first thing I want to do right now is recluster my SNPs. So you can see in the graph right now that all these have been grayed out, all those bad samples. So to recluster my SNPs, I press this button here that says cluster all SNPs. It's like the three colored ovals or circles here, and by clicking on this, it will ask me if I want to recluster. I say yes. This is going to take a little bit. Why do I want to do that? Well, I want to do that for the next steps because I don't want to take those samples into account when clustering, because as I've said, they do influence your clustering, and they might actually make some samples look, or some SNPs look as failed rather than working. So this might take a little bit, but while that is running, I'll go ahead and start talking a little bit about the sample ploidy. What is ploidy for the people who don't know? Well, apple, cherry are diploid, which means they got 2 full chromosome sets, one from mom and one from dad. A polyploid has more than 2 sets, so you can have triploid has 3 sets, tetraploid has 4 sets, and so forth. In apple, we've also encountered some aneuploids. In that case, it looks mostly like a diploid, but one or a few chromosomes either have a set missing, and so I have only 1 set, or they might have more sets. So in this case, here's some examples. You have the red and the yellow chromosomes have 2 sets, but the purple only has 1 set, and in the other one, the purple has 3 sets. Now, they lead to different intensity patterns, and I'll come back to that in a bit. Also, a lot of the error detection that will happen later on is, at least in the software that I'm using, is expecting diploids. By not having diploids, I'll get a lot more errors for those individuals. And so, well, in Apple we don't have a lot of these, so I removed them. But I mean, if you really want to keep them, I would not use them throughout the workflow. I would use the diploids if you're working with a diploid species. I would keep the diploids in through the workflow, and then later go back to your polyploids, aneuploids, and work with them afterwards, after you know which SNPs work, etc. So we like to identify them, and so how do we do that? Well, it is actually the same as what we did when we looked to sample quality. We create those histograms for the B allele frequency, and so I'll show you one again. Okay. So if this is done, you can say that yes, you want to update the statistics. That should go relatively fast. And then it's going to probably ask to do the other 2, and that should go also pretty fast. Anyway, I'll show you on the PowerPoint now. So if you looking at the B allele frequency, I told you we have 3 peaks: one for AA, one for AB, and one for BB. Now triplets, they're not AA, they're AAA. Well, that's still only A, and they're also BBB. That's still just B, so they still end up at 0 and 1. But their heterozygous genotypes are not AB, they're either AAB, and then they actually have slightly more A than B, so they shift a bit away. So they have about 33% B and 66% A, or they're ABB, and in that case they have about 33% A and 66% B, and so they move a bit to the right. So your heterozygous cluster is split up into 2 peaks. So by looking at those graphs, you can easily identify triploids. So we don't have any in cherry, but I'll show quickly again how you get there. You click on the histogram plot here, then you click on on any sample to make sure you can get to the B allele frequency, and then you click on the sample again. And I can tell you, if you're just scrolling through this and suddenly a sample appears with 2 peaks rather than 1 peak, you'll very quickly see that, and so at that point you know you have a triploid. Again, we don't have any in the cherry data set, so I can't show you any in Genome Studio itself. We've also seen something that might have had in Apple that might have had 5 peaks. It might not be as clear as this, but I'll show you another method to find polyploids, and then at that point it were clearly 5 groups, which might indicate that that was a tetraploid, or it might just really be 2 samples that got mixed more or less equally, which would also lead to this kind of pattern. And then we have aneuploids, and they are harder to identify with this method. So if we from a different way, and I'll come back to it. I know this sample is an aneuploid, and by looking at that, I see that there's like small little shoulders on the main AB peak. So that might be an indication that your sample is an aneuploid, but it might be much harder to identify that by just scrolling through these. So what is that other method I've been talking about? I'll first show it on this in the PowerPoint. This method, we do something very similar to the histogram. The difference is that we now, rather than making a histogram, we plot where each SNP is in the graph. So a BB SNPs are normally plotted on the top. AB SNPs are normally plotted around 0.5, and AA samples can be found, or AA genotypes can be found at around 0. So for a single sample, we plot where it can be found in each of these graphs, and we not just plot it, we plot it, in this case it's an apple individual. We plot it along the chromosome, along their position, so you can more or less distinguish 17 groups here, which stands for 17 chromosomes in apple. And so in this case, it's clearly a diploid. In this case, we can clearly see for the heterozygous group 2 clusters, so that would indicate a triploid. And this method not only identifies diploids from triploids. I told you before we found some things that look like a tetraploid. Here it's already a bit clearer that we have 5 different groups. So this might be a tetraploid, but again, it might also be a contaminated sample. It's not only showing us ploidy, it's also showing us poor quality, because if something is poor quality, it kind of looks like this. It's all over the place. And we also see aneuploids pretty well, because by splitting it out by chromosome, we can see that in this case, almost all chromosomes had a diploid pattern except this one chromosome, which I think is 4. So chromosome 4 showed a triploid pattern, so that would indicate that this individual has 3 sets of chromosome 4, but otherwise is a diploid. So how do we get to those plots? Well, I'll show you in Genome Studio. The first thing I want to do is we're gonna do some filtering in Genome Studio and remove some SNPs from our data set to make these plots. Why do we do that? Well, if I don't, then all my plots will look like this, and in this case we were talking about cherry samples because we have 8 chromosomes. But they all look like this, and we don't see much. And why is that? Well, if we look at the histogram plots in Genome Studio, we saw that there might still be a very small amount of SNPs that have somewhere in between B allele frequencies, something around here, and if we don't really notice them in a histogram function because all the other numbers are so high. But if we plot every individual SNP, you get a dot everywhere, and then the top parts and where we are expecting the clusters to be, like in this graph, they're probably much denser in this graph than the in-between ones, but we can't see that visually. So to get those SNPs out of there and not influencing our graph, I'm going to do some filtering in Genome Studio from the SNPs and create a subset. So I've already gone over this about excluding samples. If you now look back at this graph, I don't know if you remember, but when these samples were not included, this purple circle here was much bigger. And one of the things I'll be selecting for is the width of the cluster, so which is represented by this purple circle. And so if we hadn't reclustered all our SNPs, this SNP might have been removed, even though it was a good SNP. Why do I remove SNPs that are really big, with a big circle? Well, that is because most of the times actually those SNPs have multiple clusters in one genotype group, multiple groups. And that might indicate that SNP is actually targeting multiple loci, or there might be some other things off with that SNP, so I don't really want them for this graph right now. So that's one of the things I'll be filtering on. So how do I filter in Genome Studio? I'm gonna see if I can make this a bit smaller and make Genome Studio a bit bigger. Okay, like this. So to filter my SNPs, I'm gonna go to the SNP data, SNP table. So on the right pane here in Genome Studio, standard default data table is open, but as I showed you last time, there's also a SNP table tab. So I'll click on that. And then this table holds all the parameters that I want to filter on, so that's why I go here. And then I press the filter button, which is this button here. And then I start filtering. And I'll go through the process here. So I want to filter, the first thing I want to filter on is the call frequency. The call frequency, as you remember, is saying what proportions, in this case, of individuals is called. So when we were talking about the SNP table, the call frequency, what proportions of SNPs is called or has a genotype for this individual, whereas the call frequency now, because we're in the SNP data, in the SNP table, is what is the proportion of individuals that had a call for this SNP. And so I want to filter on this, because if that is really low, it just means that something is probably wrong with my SNP, and then I don't really need it, at least not for this graph. So a good value would be a call frequency larger than or equal than 0.9. And then to add this to the filter criteria, I have to press the arrow here, and then it should appear here in this window where it says filter tree view. The next thing I want to filter on is the minor frequency, which is saying, so if a SNP is called and it's and you have certain individuals are AA, certain individuals are AB, and certain individuals are BB, it might be that either the A allele or the B allele is occurring less frequently. So there might only be one individual that is a BB or an AB, and almost everything else is AA. That would mean that the B allele barely occurs in my germplasm, and so the minor frequency is a measure for how much that the allele occurs that is the least present. And so if the minor allele frequency is 0 or really low, it might actually indicate that you have a monomorphic SNP, which means that would be only AAs or only BBs, and so there's not really a polymorphism there. And so I don't really need those in the graph, so I'm going to remove those. So any SNP that has where the minor allele frequency is more than 1% is okay. So I add that to the filter again by clicking on this arrow here, and as you will see now, automatically this end node appears, which means that it needs to be end this, and that if right now we want end, if at some point you're doing some filtering and you don't want end, you can right click on this, and by right clicking you get a menu where you can choose other operators such as OR, and then that will change to OR. Then the next parameter I want to filter on is the GC score, and the GC score is a score on how well your SNP got clustered. So individuals here that are really close to the center get a high score. These one would get the ones that are really far off get a really low score, and anything in between gets an intermediate score. And this 50% GC looks at all the individuals, at the score for all the individuals of a single SNP, and it looks at all the scores of the individuals and it chooses the median. And so the higher that median is, that means the more individuals it got clustered pretty accurately, and the lower it is, it means the more individuals didn't get clustered really well. So it's a quality score, and so again, I only want to have good SNPs for making these plots. So that's why I'm filtering on this one, and a value of 0.4 is a good value. By the way, the values that I'm using, the ones that are also in the table here in the presentation, they're not just chosen at random. They're based on experience, and I've done a lot of analysis to see what values are good and what are not. I don't really have time during the session to go over them and why, like how I came to these values, but these were determined for the 8K apple, and they seem to be holding up for the 20K apple SNP from Illumina and the cherry arrays. So they give a good subset of SNPs to work with and to get to these figures. The next is the score I want to use is the GenTrain score, and the GenTrain score is here in the subcolumns, and just as before, this is grayed out, and that's because the GenTrain score and the cluster SEP and some of the other ones that we've been using are actually parameters that are part of our array, and our array here is listed as RosBREED cherry 15K. And I don't know if you remember, but last week we went through the sample sheet file and we had to say the manifest, and as we were loading all our samples into Genome Studio, we had to show where this manifest file was stored. So this is the name of our manifest, and so if you use a different array, you'll have a different manifest, but that manifest name will be here, and by clicking on that, you make all the other subcolumns available. And so then I want to choose the GenTrain score, which is an overall quality score of the clustering, and I want to say that it's larger or equal than 6.65, and then I add this one again to the tree, and you see that this one is getting bigger and bigger, and it's becoming harder to see everything you've chosen, but it's also listed here at the bottom of this window, the ones you have chosen. And then I also want to use the cluster SEP, which is yet another quality score, and this is a quality score for how well my 3 clusters are separated. So in case they are like in this graph, like here, the cluster SEP is a pretty high value. If all the clusters are squished together, as I've shown you in some graphs before, then that cluster SEP value will become lower. And a good value for this is 0.4, and again I add this by clicking on the arrow. And then there's a few parameters that I will use to lock in the position of the SNP. As I've said before, we don't really want any SNPs which have clusters in this region or SNPs that have clusters in this region. So we do that 2 ways. One, by saying, well, the clusters mean, like the central position of the cluster should be here, more or less here, or here, so at 0.5 or 1, and we allow a little bit of deviation. The second thing that we do is by saying the clusters can't be really wide, because even if the average position is here, but there's a second cluster here, we don't really want that. Let's say that there's a big AA group or cluster with 2 groups in it. We might still have our average on a acceptable position, but we still have that extra cluster. And to avoid this, we look at the width here of this cluster. So those are these 6 values. The AAT mean, so T stands for theta, which is the x-axis. So the mean, those 3 means show where is the center of my cluster, and the depths are how wide is my cluster. So I'm gonna go a bit off of the order of the table, and that is because I have this larger than selected right here, and I'm just gonna keep that as long as I can. So the ABT mean needs to be at positions larger than 0.375, which means that we don't want the AB cluster in this part of the graph. We want them far enough to the right. And similarly, the BBT mean needs to be high enough, and in this case it would be 0.875, which means that we want our BB cluster, or at least the center of the BB cluster, in this area of the graph and not too much to the left. So I'll add that too, again, by clicking on the arrow. And then I think all the other parameters require smaller than or equal, so I'll change that now. And so for the AA mean, we use 0.125, so we want it left enough of the graph, we want it somewhere here, we don't want it too far to the right. And we've already selected on the AB, on the mean position of the AB cluster, but we only told it to be not too much to the left. We also don't want it to be too much to the right, so we need to add a filter for that, which is 0.625, and we add that. And then now we can go and add the filters for the width of the clusters. And so for the AA and BB clusters, I don't want it to be, the deviation to be larger than 0.028. So I'll add the AA. Given that the BBX TDEF has exactly the same values, I'll first go to the BB TDEF and add that one, so I don't have to change anything. And then for the AB TDEF, well, with the AA clusters and the BB clusters are expected to be at 0, but some individuals may deviate a bit because there's some noise for the other allele, and so they vary a bit. For the AB cluster, that noise can be either direction. Also, with the AB clusters, we expect a 0.5, but that would mean that each allele, so the two alleles that are present, bind equally to the probes, which means exactly 50% of the probes that get bind get the A allele, and exactly 50% of the probes that bind get the B allele. Well, whichever allele binds is a little bit a chance, so there's also a little bit of deviation there possible because of that. And unlike the AA cluster and the BB cluster, the AB cluster, these samples can deviate in both directions. So I'm already expecting that the deviation can be larger for this cluster than for the other two, and that's why we also allow a larger deviation for this cluster, and we allow actually the double that what we allow for the AA and the BB cluster. So if we've added all these filters, here we can go ahead and press OK, and if everything went well, and it did, for me at least, we see here that we have about 2076 SNPs still chosen after our filter. So that's a big cut down from our 13,500, but that's still enough to make our graphs. So now that we've done the filtering, we can go back to the full data table by clicking on that tab here. And as you will see, or as you can see, that also this table has now been filtered, which is really great, because otherwise we had to export both tables and then combine them in Excel and do all our filtering in Excel. But because both tables get filtered the same way, we don't need to do that, and that saves us time. Now I want the B allele frequency for this, and if you look at this table right now, there is no such thing as a B allele frequency for a sample. It only lists the genotype, the score, the theta, and the R, and as you will see, these first samples have all no calls, and that's because those are all the samples we excluded, so they don't get a genotype call. So to get the B allele frequencies in here, we have to go and change the layout of our table, and we do that by clicking on column chooser here on one of the top options is the one with the 3 blue bars, which is column chooser. And so by clicking on that, we get this new window where we can decide what is in our full data table. And so as you can see here, there's a bunch of parameters that it says that are displayed, and also subcolumns, which means that for each of these samples that it is displaying here, it will display all of these values. Well, I don't need all of that. So what do I not need? Actually, I don't need the name of the SNP. The index is enough, and I've mentioned last time why why I rather use the index. It's a number. It's pretty short. And it's consistent as long as you're working with the same array. So whereas the names are also consistent, but they're much larger and sometimes harder to work with. So the index is a good one to work with. I don't need the address either, and so I'm holding Control so I can click on multiple columns or on multiple values here. I do want the chromosome and the position, because remember, we're going to plot it against the SNP's position. I don't need the GenTrain score anymore, because we've done our filtering on that. And I don't need these values. So I've been holding Control while I click on all of these values, and then I can click here on Hide, and they move over to the hidden columns set. So that will mean that they will be no longer in our table. I also don't really need genotype, score, theta, or R, so I can highlight all of these by just either holding Control and clicking on all of them, or clicking on the first, holding Shift, and clicking on the last one, and then say Hide. And that will move all of them also to the hidden columns, but in this case the subcolumns. I do want the B allele frequency, which is right now is under the hidden subcolumn, so I want to click on that one and then say Show. And so by doing that, I've now changed the layout of my table, and then if I click on OK, you'll see that all these columns disappear, and now we have the B allele frequency for all our samples. So now I want to go and use that to make those graphs. I've not found a way to do that in Genome Studio, so I'm going to do that with R. So first thing I need to do is get this table out of Genome Studio, and I do that by clicking the Export the table to a file, which is the third button. It's the sheet with an arrow going out. And so if I click on that, it will ask me where I want to save this. I made a new folder that says Genome Studio Output, where I am saving some of the output of Genome Studio. You can save this where you want, but please remember where you do. And then in this folder, it's asking me whether I want it tab delimited, which would make it a text file, or I want it as a data file, and I'll choose for a CSV file. That's slightly easier to work with, and that's also what R will be expecting. And then Genome Studio always saves your data tables as a full data table name or as a SNP table if you save the SNP table. So you want to give that a more meaningful name. So in my case, I will just call it 6 plus 9k and then B allele, because then I know that this is the file that holds all my B allele frequency for all my samples, and then I hit save. Then I'll get a question whether I want to only export whatever I have selected right now, or if I want to export the whole table, and so I say yes, because I want to export the entire table. I want everything. This should go relatively quickly, and if you then get asked if you want to view the file, you can go ahead and click yes, because there's a few more things we need to change. So if I click on yes, that should open the file in Excel. So here we go. And so there's a few things I would like to change in this file before I create the graphs. One is that if you look at the top row, it is listing all the individuals. But it's not just listing the individuals. It has the name of the individual, but it also says .b allele frequency. It does that because if you export multiple parameters for Sweetheart, for instance, in this case, you need to know which is the B allele frequency, which is, for instance, your genotype. And in some cases, it's pretty straightforward. In other cases, it's not. So it's telling us that this is a B allele frequency for this sample. Now, everything is B allele frequencies. So I don't really need that anymore. And the way the R script works is it will take this value and use it as the title of your graph. So I only need to know which individual this is. I don't need to. I know it's the BLE frequency. So to remove that, in my case, Sweetheart is the first one, and so I take the first sample, whichever one you have there, and I hold Control Shift, and I press the right button, which will then select everything on the right of the first sample, and then I press Control H, and by pressing Control H, I get the find and replace option in Excel, and so I will write here now dot B allele frequency or frec, because that's what says on top. It's not case sensitive, so you don't have to worry about the capitals. And then you say replace with nothing. I just want them to be deleted. So if I replace this with nothing, they will be gone. And then I hit replace all, and then it will inform me that it made 77 replacements. And then if we look at this now, it just lists our individual's name rather than all that other stuff. Now I want to go and plot this towards their chromosome position, and to do so, I would like to have a single value that both holds the chromosome number and the position within the chromosome. So to make this new number, I'll click on the first column that holds a sample. This should be the D column if you followed all the steps as I did. And you right click on the column and you click Insert, which will give you a new empty column. Now before I can do anything, I also need to you have a number for the chromosome, and right now many of the chromosomes are not a number but are a word. So I'll show you what all our options are here for chromosome. So for chromosome we have the numbers 1 through 8. In Cherry there's 8 chromosomes. Then we have PAV underscore 1 through 8, and we have a few that are PFR. So I think I briefly mentioned this last time, but the cherry array is made for both sweet cherry and tart cherry, and tart cherry is a tetraploid crop which has 2 subgenomes, the avium subgenome and the fruticosa subgenome. So these PAF and PFR is indicating which of these 2 subgenomes it's been targeting. Not only that, sweet cherry is Prunus avium, and so everything that's PAV is actually a target for sweet cherry, and we figure that it will also work for the avium subgenome of tart cherry. So this is really useful information, but not what we need right now. So right now we just want to have the chromosome number, and it doesn't really matter which subgenome or it is targeting. So I want to get rid of that, and the easiest way is again with find and replace. So that was pressing Ctrl and then H, and that will bring up this window. And so this time I want to remove all the PAV underscore, and I'll say replace all. Make sure you have the B column highlighted when you do that. So if you do that, it will make about 1,000 replacements. And then the same goes for the PFR, and I can say replace all. Now depending on the germplasm you've been working on, if you use this sweet cherry array, sometimes it will keep some other SNPs that set mitochondrion or chloroplast, because we added some of those SNPs on the array as well. And they're not really useful because they will not show anything about ploidy. So in that case, you might want to just delete those rows off first before we continue. But normally, in this case, they're not here. So we don't have to worry about them right now. So next thing I want to know is, what's the size of my chromosome? What's the highest position I get in a chromosome? And there's several ways to do that, but one is by having this filtered here, and then looking at what the values are, and we see that the highest value is about 47 million base pairs. So there's nothing larger than 47 million base pairs. So I could theoretically say first chromosome goes from 1 base pairs to 50 million base pairs, and the second chromosome goes from 50 million and 1 base pairs to 100 million base pairs, and so forth. That works, but that might be a bit harder to trace back which chromosome is what. So what I'll use, rather than a value that's still use a value that is higher than the largest position, but I will use a power of 10. And so in this case, if it's about 50 million, then the closest power to 10 that is higher than that is 100 million. So at that point, I will say my chromosome 8 is, I will use a function here that uses the chromosome position and makes it a really high number, in this case a multiple of 100 million, and then add the position to get unique values and to make sure that positions on larger or on different chromosomes are on different positions, and that the higher the chromosome number, the higher the position is. So to do that, I start a function in this new column that I created here. And I say, to do so, I start with equals, which starts the functions in Excel. And I say, well, I want my chromosome number, and then I want to multiply this with a high enough number. So as I said before, in our case, this is 100 million base pairs. So you can use there's a function to do a power in Excel, but you can also just type 100 million, and so that would be 100 for 100. Then 3 zeros, 1, 2, 3 to get to 100,000, and then another 3, 1, 2, 3 to get to 100 million. And then I want to add the position of this value, and then I press enter, and so then I get a new position that I will be using in my graphs. And then by double clicking on this little square here in the lower right corner of the cell, it will automatically fill everything for all my SNPs. Now I want to get rid of this formula because I'm going to delete those two columns here, and if I delete those two columns and the formula is still here, then I will lose the value. So to get rid of the formula here, what I do is I highlight the first cell, I hold Ctrl Shift and press down, which selects everything. Then I press Ctrl C to copy everything, and then I right click on the first cell, and that gives me all these options here, and there should be an option saying values. It might look slightly different in your version of Excel, but there should be an option that says values, and by clicking on that, all of these become values rather than their formula. And then I can go and safely delete off columns B and C, which hold chromosome and position. And so this column doesn't have a name. It doesn't really need to have one, but I'll give it one anyways so that I still know what it is if I ever come back to this file. And so now your Excel file should look something like this, where the first column holds the SNP name, the second column holds the position, and all the other columns hold the B allele frequency and have the name of the individual on top. And then I can go ahead and save this file again. And so now I'm going to use this file in R to create all these plots I've been showing you. And so to do so, I am going to go to where I saved that Excel file, that table from Genome Studio. And so in my case, that would be in Genome Studio Output. So I select this file, and I'm going to copy it, and you can copy it by hitting Ctrl C, and then I'm going to go to where I have saved my R scripts. And so if you have just copied everything from the Dropbox folder, that would be under Software, and then R scripts. And as you can see, there's already some things here. I'm going to delete them and hope that that the file still copies, and it does. So now I have my file copied together with the R script, and the R script we'll be using is the B allele analysis. So if you have installed R and you have installed Genome Studio, this file should be opened with Genome Studio by double clicking on them. Should it not open with Genome Studio, you can make Genome Studio your default belief by right-clicking on it. It should give you an option to open with, and then it would give you a lot of apps. Hopefully RStudio is in there. Then you can select RStudio, make sure this option is checked where it says always use this app to open R files, and then then your default becomes RStudio to work with R scripts. So if we look at this R script, so R uses code to do some stuff, but even if you don't know anything about R, especially this script is really easy to run. There's a few things you need to know. This in RStudio, what is here in green is that green means the text, and right now this first line of code is saying read my data file, and my data file is has a certain name. Now this name is not the name that I gave my file, it's the name I gave some other file when I wrote the script. So I want to change this name to the name that my file actually has. Now I can just type this in here and hope I don't make any typing errors. I can also go back to my file here, and if I right click on my file, I get an option to rename my file. So if I click on rename, it will highlight the full name of my file, and then I can just press Ctrl+C to copy that name. After that, just click anywhere to get out of changing the file name, and then here in RStudio press Ctrl+V to paste, and then I get the file name. This file name was rather easy. If you have more difficult file names, that might be a good trick to get your file name correctly copied somewhere. Really, you don't really need to know much more about this R script other than here where it says PDF, it will save all our plots in a file called BLEL PDF. So if you want to have maybe say this is the 6+9 K BLEL, you can do that. So you can change the name of the output here. Then we need to tell R where to find our file, because it's not always looking standardly. It doesn't know where to look for immediately normally. So we need to change our working directory, and our working directory is basically telling R, this is where my files are, this is where you have to save the files that you're generating. So I do that by clicking on session here, going to set working directory, and I can say to source file location. What that will do is saying set the working directory, which means look for files and save files in the location where this R script is saved. So by clicking that, you'll get in the bottom here in the R, it's saying my working directory is now this. So again, that's under sessions, set working directory, source file location. If you want a different location, not where your R script is, you can say here choose directory, and then you'll get a pop-up window that will let you choose any directory. But right now I've copied this file in the same folder as the R script so that I can just say source location. And then we can just, if everything goes well, we can just select all of this code and hit run, and it should do everything. It should load our file, it's creating a PDF, and then here this piece of code is just saying plot now the second column versus the BLEL frequency. And so that's why we had to have our position in our second column, and it will use the first very single value in the top row as the name, where the name of the individual was as the title. So if that all went well, you should get now a PDF file here with the name that is specified in the R script, and by double clicking on that, just give it a second, you'll get this, and you might be like, I thought we were going to see 3 nice clusters. Well, yes, if you go to the next page in the R script, in the PDF, you'll see starting having nice clusters. The reason we don't see those in the beginning is because those samples we excluded still had their BLEL frequency exported, And as I had sorted them in a way that they came up, the way I had sorted them in the SNP data table, in the sample table. So the way I had sorted them here in Genome Studio in the sample table was that they were on top, and so because they were on top here, they became the first here in the full data table, and because they're the first here, they're also the first that get their plot made. And so you can see clearly here that those ones that we set were no good are no good. And then as we go further, you can see that yes, everything looks really nice. And so, well, we don't have any aneuploids or triploids in this data set, so there's not much to show. They all look really nice diploids. The only thing you can see here a little bit is that there's not that many AB SNPs for this individual. There's still a few. I don't know the pedigree by heart of this one. It might be that this one is a self, although I think then there would be even less heterozygous ones. It might also be that the two parents are pretty related, and so that for this chromosome, it became really homozygous because the parents share a good chunk of their chromosome. So that creates all these figures. I think that is it for all the B allele frequency and the ploidy to check. Yep. So that's all of that. So now we have a good idea of which samples to use, which samples not to. There's one note I have to make. We have seen in Apple one or two segmental aneuploids, which means they're not really an aneuploid. They just have a large chunk of one chromosome missing for one reason or the other, and really we don't really identify this with any of these methods. We'll only see that later on during the error detection. I'll come back to that. So the other thing I wanted to talk to you about today is getting a good SNP set. I'm gonna skip these slides. If you have time, check them. They're just showing what kind of misclusters there can be, and it's kind of showing how you can solve these, and that is by looking at parents and the segregation in offspring. You can get an idea of what's truly happening with your SNPs. But the main point I wanted to make here is that there's several SNPs actually that might work or might not work, but they do lead to errors in their genotyping, and you want to identify those. And so there's actually two approaches. One is using filtering SNPs, as we've done before in Genome Studio today, to get a subset that subset works to get good SNPs. Unfortunately, the way we've been filtering it also means that a lot of SNPs with useful data are lost. There's not really any consensus in literature, or there's no guidelines from Illumina how to filter really. There's some parameters like the Gentrain score that get used more than others, and there's some ranges in values that are being used. So if you want to do that, you can do that. The other way to do it is a software called Assist, and it's made to choose a subset. It was developed through Frubidomics for the 20K Apple SNP array. And what it does, it can identify null alleles, or if there are multiple class AB groups or multiple AA groups, it can distinguish those properly and also give them another genotype. So to get Assist running, we need a few input files. So one of those input files is called the final report. We need to make that one, and we make that one in Genome Studio by going to Analysis on the top, then choosing Reports, and then choosing Report Wizards. So if you're there, so under Analysis, Reports, and then Report Wizards, and then by clicking on that, you'll get a few options to what kind of reports you want, and the first one we want is the final report, and that's the one that is selected already. So you go ahead and click next. You get the question, which chips, which arrays do we want to use? Well, we want to use all of them. We want to use as much data as possible to identify SNPs that work and that don't work. So go ahead and click next. Then it's asking us if we want to have the samples that have been excluded, if we want them in the report or not. And we don't want them in the report. As I said, those can influence the clustering, so we don't want them. So any individuals we removed need to be removed from the report. Then it's asking me about whether I want to exclude some SNPs, and I don't. I want to include all SNPs. If you are using an older version of Genome Studio, you might get a question somewhere along the way whether you want to redo some statistics, and then the assist manual says you have to choose the 10th percentile GC score. So if you get that question, which everybody should have the latest version of Genome Studio, because that was what was given in the Dropbox. You probably don't get that question, but if you already had an old version of Genome Studio installed, you might still run into that. So if that was the case, you need to choose the 10th percentile GC score. Okay, and then we get to this window where it's asking us what we want to have in the report. And there we have to make sure that standard is selected. So we want the standard format of the report. And then we want to have all these displayed field columns that are already by standard here: SNP name, sample ID, allele 1, allele 2, and GC score. But we want assist requires some more. And so the ones it requires are GT score. And so click on that one. It's somewhere in the middle of the list, and you click on show. And then assist also wants theta and R. So it's basically reporting where each SNP each individual in the graph is. So you click on theta, show, click on I want to show R. And the other thing that is important is that these are in this order. So sample name, sample ID, allele 1, allele 2, GC score, GT score, theta, and R. So not in any other order. Also important here is to say that we want it grouped by SNP rather than grouped by sample. Everything else should be the default. So this needs to be on tab, set that it's a tab rather than a comma separator. But that's normally already chosen by default. And then the other thing it will ask, or Genome Studio will ask, is where do I want this saved? And so I want this saved in my Genome Studio output folder. You can save it wherever you want, as long as you know where you save it, because you'll need the file. So this folder is okay. And then it's asking me for a report name, and I think that Cherry 6+9K final report is a good name. It tells me that I'm dealing with this array and that it's the final report, so I know what this is. So then I go ahead and click finish, and that's going to take maybe a minute or so to write the report. So I'm going to wait for that. There's 3 more files that we need to create. I guess we can start with one that we don't need Genome Studio for while we're doing that, and that is in the PowerPoint at least is the 3rd file, which is the pedigree file. And so the pedigree file just lists each individual and what its parents are. So if you go to the easiest way to make this file is by going to the sample sheet file that I've shared with you all, because if I open this file, you will see that it has a sample ID and it has the 2 parents of this individual. So I'm just gonna copy those into a new sheet for now. So I'll just make a new sheet here. It's the quickest way. And I'll select all my individuals. So I click on sample ID, I hold Ctrl Shift and I hold down, press down, which then selects all the individuals. And I press Ctrl C to copy it, and I copy this into a new sheet. And then I need the two parents. So I'm gonna just select the two headers or the two names of that column, and then I hold Ctrl Shift down again, and it's gonna require a few more presses down to get to all the individuals, because every time there is a blank it will skip it. And so at some point, if you press too far, it will go all the way to the bottom of your Excel file. You don't need all of that, you just need the two that are the last. So you can go back by pressing up if that happens to you, and then you press Ctrl C to copy it and Ctrl V to paste this. And so now we have here every sample that is on the array together with its parents. And the parents, if you listed the parents and the parents are not in part of the individuals, that's okay. So don't worry about that. Then the only other thing that assist requires is that you use different headers, different names for your columns. So the first one that you need is has to be, what is that, backslash. So to be able to put that into Excel, you have to double click on the cell so that you can edit it. If you just start writing this backslash, then you'll get something else. I think then you get every tab gets assigned a letter, and then you can use that letter to open that tab. But that's not what we want. We want to actually write the backslashes here. So you say backslash sample ID. I believe assist is case sensitive, so make sure that sample, the S, the I, and the D are all capital. And then the first parent should have as a header mother, and the second should have father as a header. And so Excel doesn't expect assist doesn't expect an Excel file, it expects a text file. So to get this to a text file, I select the first cell, the one that says sample ID. I hold Shift down and Control and press right, so that makes me select all my first three cells. And then if I select down, it will select everything. I hit Ctrl C again. I make sure I open Notepad. I start a new file in Notepad. If you already have a file open here, you can just double click like you would doing, for instance, Google Chrome or another web browser, and it opens a new file for you. And then you just paste everything in and you hit the save button, and then it's going to ask you where to save it. And I am going to go put this for now in the Genome Studio output file, even though it's not directly a Genome Studio output. I'm gonna just have all my files in the same folder for now, for today, and I'm gonna say this is this 6 + 9 9K. This is the pedigree file, and I'm gonna save it there as a text file, and that's good enough. And so now Genome Studio should have by now made your report. So if I go here, you don't need to view the report. It should have been made. Well, sometimes if you have a lot more individuals, this report gets really big, and it might slow your computer down. It might even make Excel or so to stop working. So then the second file that was listed in the PowerPoint was where is it? Is a DNA report. So to get the DNA report, that's also from Genome Studio, we go to Analysis again, Reports, and Report Wizards, just as we did before. And then if you are in your Report Wizard, you'll see that there's also an option for DNA report. So go ahead and click that one. Then go ahead and next. We still want the data for all the arrays. We still don't want any excluded samples in our report. We still want all the SNPs. And I believe we have to group it by sample ID. Yep, so we have to group it by sample ID, so the default is okay. And then it's asking us again where we want to save this. Again, Genome Studio output folder is good for me, so I'm going to save it there together with all the other files. And this name works for me because it tells me what array it is and that it is a DNA report. So I go ahead and click on finish, and this should go much faster than the previous file. Still don't need to see this. Now, the more samples you have, the longer that this will take to make this file. But given that we only have 77 samples, it went pretty quickly. The last file we need is a map file, which is optional. So the map file is optional, and ideally when you use it, you can use genetic positions rather than physical positions, because Assist will add that position to some of its output file, which can be really useful later on. Right now we don't have any genetic positions right now. We only have physical positions. If you want the physical positions exported, well, we want them for all the SNPs, not only for the ones that we have filtered. So if your filter is still on in Genome Studio, then you click here to have it removed, and so that you get all the all your SNPs again. Then we export this table again, which is using the third button. I'll just say that's 9. Okay, array here, and I'm just going to call this positions .hex because I really want just the positions. And I guess I'll save it as a CSV file because that's easier to work with. Again, it's asking me if I want the entire table, so I say yes. Go ahead. This should hopefully don't take too long. But that would give me all the positions. Actually, the other way to do it, actually, which would have been probably the smarter way, I'll show you once this is done, is to highlight both of these tables. Sorry, no. If we want to do that, we have to go to the column chooser again, because Assist requires us to have the name of the SNP, not the index. So we need to make sure that the name is shown. So again, here on the top there was this 3 blue bars. This is the column chooser. I click on that, and then I choose name. I choose show. So that should make name come up, and I can bring this all to the top if I want. I'm not sure if I don't do this, if it wouldn't show at the beginning. So I'm gonna make sure it's at the top here so that it just shows at the beginning of my file. So I make sure name is back in here, and then I would have to export the data table again because I do need these. But the quicker way to do it is, rather than exporting the whole table, is by choosing the 3 columns, then choosing the first sample, or the first SNP, go all the way to the bottom of the file. Hit Shift, hold Shift, and click on the last sample. So now all of the SNPs are selected. Our 3 columns are selected, and I can now press the second button, which are the 2 sheets, which says copy, and I'll just go to any Excel file I have open right now, create a new sheet, just so that I have a blank page, and now I can hit paste, Ctrl V, to get all of my SNPs and all of my positions. And then we have to still do the same as we did last time. We select the chromosome table, we press Ctrl H, we want all the PFRs gone, so we want PFR underscore replaced with nothing, so we get a full number, we just get the number. We want the same for all the PAVs underscore, replace all of them. And so this time, if I will show you by filtering this for this column, you can see that we still have chloroplast and mitochondria here. So I want also those to be changed to a number. So I'll press Ctrl H again. I'll change this to chloroplast. And I'll say chloroplasts are chromosome 9, because we have, well, in cherry we have 8, so the first available number is 9. So I say replace all. You can see that there's 27 of those on the array. And then for mitochondrion, I'll use 10 so that I still keep track of what is what. And I say replace all again. And then so now everything here should be a number in this column. And so we have chromosome position and our SNP name, which is what SYST asks for a map file. And then it does require us to change the headers as well. And so it's, in this case, it's \\SNP in capital letters, ID small letters. The second one is chromosome, just with a capital C, and then the last one is position, which is already is. So that would be our map file, but again, SYST wants a text file, so we'll select everything. We just take the cell in the top corner, hold Ctrl Shift, press right to get all our columns, and then down to get all our rows, and then hit Ctrl C. And then if we want to make a new text file, we just double click somewhere next to these tabs on the top, which creates a new file here. Paste everything in and press save again, and this time I'll call this 6+9k map and save it. So that should be all our files for SYST. I know that we're going a little bit over time. I'll try to be quick here. This should all SYST can be found under software. It's one of the softwares that you just have to copy to your PC. So in my case, it's still in that folder. Then you have the SYST folder. If I click on that, you get the SYST version 1.1 folder, and when you double click on that, you get to the files where SYST is stored and where the software is. So I'll wait a few seconds for everybody to follow. So you find the folder where SYST is, and then you go all the way to you see this, where you can find the SYST Windows version 1.1 executable. And so that's the one you want, and that's the one you can double click on it, and then it takes a few seconds. Sometimes it takes a bit longer. And this is SYST, and SYST will open. And so SYST won't let us do much other than selecting our input files. So that's where we'll start. So it's asking us for the final report, the DNA report, the pedigree, and the map file. So we'll have to tell them where we'll have to tell SYST where these files are. So for me, that's in my Genome Studio output folder. That's why I put everything there. And so the first thing it asks is the final report. So that would be this one. So you just double click on that, and then it will be here. And the next it asks for is the DNA report, which is still in the same folder. It's a CSV file. So if you named it with DNA report in the name, then it's pretty easy to find. And then you choose that one. You can double click on it, and then you will have told SYST where to look for that file. Third file is the pedigree file. That's why. So this is a 6+9k pedigree file we just made. So I'll choose that. And then for the map file, it's the same. I choose a 6+9k map file. Everything for me is in the same folder, so it's relatively quickly to get all these files. So once you've told SYST where to find its inputs files, you can go ahead and click on OK. And SYST tells us that the files are looking okay, so that's good, and that we can now set the parameters. And you see now that this button has become available to us. So we're gonna set our parameters. So there's a few things we need to change. So first of all, it asks for the population type, and the default is an F1 across population. So a big F1 family, which we don't have right now. If you do have that, even if you can load up your Genome Studio files, you don't have to make them separate for each family. You can have, I believe, all your families in one DNA report and final report, etc. Because you can here say which are the parents of the F1 family, and then assist will only take those. But in our case, we want germplasm because we have a lot of individuals. They're related to pedigree, but they're not big families. Then other than that, I'm going to have to check a little bit what our parameters are, because I don't know them by heart. Here, the allowed missing data is basically what is the call rate for a SNP that you will allow. Or the opposite of the call rate, basically the rate of missing data. So the 0.5 is a good value, and is that here in the parameter settings here? Okay, then the call rate tolerance is telling us this is for individuals, so assist can exclude individuals, and this is saying what is the average? Well, it will check what the average call rate is for individuals, and then any individual that has a lower or a much lower value, it will exclude. And here you can say how much lower before it excludes a sample. This is saying how much segregation distortion do we allow. The lower the number, the more distortion we allow. So what I do is to allow for as much distortion as possible by just typing a lot of zeros till assist stops adding zeros. Why do I want to allow segregation distortion? Well, for instance, in cherry we have the S allele, and so in certain cases we're gonna expect distortion, because if two parents are sharing a single S allele, then the father will never pass on that S allele to its offspring. So even though we would expect that father gives each allele equally to its offspring, it won't happen. So that's segregation distortion, and so we want to allow that because we know that in certain places in the genome that is happening, and we still want the SNPs for that region in the genome. Other reasons why might be that certain combinations of alleles might be lethal or sublethal, and so those allele combinations are going to occur less in our germplasm, but we still want to have SNPs for those regions. Then other than that, this is the unexpected per individual. So that means assist is going to look at pedigree, and for instance, if both parents are AA and the offspring is AB or BB, it will say that something is wrong, and so it's telling us how much can be wrong before I don't trust this anymore. And then the frequency of the rare allele is probably is one of the last things you have to set. I'm going to set it here to 0.01, which means I allow for, so rare alleles is like the minor allele frequency. I want to allow at least 1%. If you have a larger data set, you might want to allow smaller numbers, especially if you know you have one or a few individuals that are a bit special or that are not as closely connected to the rest of the germplasm, because they may have rare alleles, and so you want to still see that. So that depends a bit on your germplasm to what level you set that. And then the number of chromosomes is always 17 here because it was written for apple, and in our case cherry is 8 plus the 2 that we created for mitochondria and chloroplast. So we hit here number of chromosome will be 10. So once you set all these parameters, assist will allow you to run it. So you can go ahead and press run. I want to warn you, Windows might tell you that that assist stopped working. Don't worry about that. Assist is still working, and just if I guess I can force this, I think, by going to the PowerPoint and back to assist. So if you can see this, it right now is saying not responding. Every time when I just let it do it, keep running, at some point it will just pop up as the run is done. So Windows might think it's not running. Actually, assist is working well. As you can see right now, I got all the results in for assist. And so assist now is telling me, giving me a summary of how many SNPs it approved, which is 2,000, almost 2,200, and how many SNPs it thought didn't work. Now that's a lot. Well, you can see that a lot of them are monomorphic. So those have not shown any polymorphism in our germplasm, and we have a pretty small set of individuals, and know that if we do this in a larger set, there's about, I think, 1,000 more SNPs or so that has polymorphism, so that already increased that number. Then it's saying a certain number of SNPs has failed. The shifted homo ones are the ones where the homozygous ones are not, groups are not where you expect them to be, and so it considered those as failed. I think that there's a lot of these SNPs actually hold valuable information and can be saved, so actually I would like to save these. And then there's a certain, and so as I said, there's a certain number of robust SNPs. And then here at the bottom, it's for every SNP, it is telling us whether it's robust or not, and giving us some other statistics on it. And you can you can sort this table. And so if I sort this table, you can see here the shifted homozygous ones. Well, this is probably an AA cluster, so automatic calling doesn't work, but manual changing might work. There's actually there's normally a lot of those where that happens, where you can actually still have good results if you put in a little bit of, if you look at it manual. There is actually also some, I think, that have the where just one of the homozygous clusters is missing, and it also considers those failed, but I don't necessarily think they failed. Their automatic calling may have failed, but they hold a lot of valuable information. So some downsides of assist is, unlike Genome Studio, I cannot click here on anything and see what sample this is. I cannot really get an idea of what's going on. I cannot just select some individuals and make some manual changes. So for any step after this, once I know what the good SNPs are and what not, I'm going to go back to Genome Studio, because I can do this in Genome Studio. The other downside of assist, I think, is that you cannot save your results. Once you close assist, you lose you lose it, and you have to run it again. Now assist is consistent in what it reports as long as you use the same files, but there's no way to save your project. So to save all the things you did, you can go to export, and it gives you some export options. The summary that it exports is this table. The custom genotypes is for all the SNPs that assist approved. It will tell you for each individual and each SNP what the genotype is. The SNP information table is this bottom table here. The Mendel error report tells us where there were inconsistencies between offspring and parents. I don't need a join map file right now. I don't really need this. I don't need the Mendel error report, because I'm gonna do that a different way. I don't need join map, but join map would be useful if you're making genetic maps. I do like the FQ data prepper. FQ stands for FlexQTL data prepper, so that's the software we're going to use. So yes, I would like my output as that, and Plink is another software we will use. So yes, I want some input files for that as well. And then Assist can also make structure files if you want those. Then it's asking you where you want to save your files. Right now it's again saving it to the Genome Studio output. You might want to make a different folder for that. And then if you press the output file prefix, you can give this a name, and all the files that it creates will start with that name. So if I say 6+9k underscore, then all the files Assist creates will. So if I click OK, Assist should make all the files, and then it says that it did so successfully. And as the last thing, I'll show you that it did so. So for me, it did it in the Genome Studio output, and you can now see that there's here a lot of the 6+9k underscore, which is the name I gave it, and then the rest of the name tells us what kind of file this is. I guess I'll stop here. Next time I'll show you how we can get some of this data back into Genome Studio, because that can be pretty useful too. So yeah, that is it for today. So as a reminder, today we checked for sample quality, and now with Assist we've gotten a subset of SNPs that we know is reliable, or we know at least how our SNPs overall are performing. And so next time we'll use this subset of SNPs to go in and look at pedigree information and make sure our pedigree information is what it should be. So thank you everybody for joining. If you have any questions, please let me know. Feel free to email me. And if you know of a good way to share the videos or the recordings, please let me know so that I can make sure that you all have access to them. Okay. And unless if there are any other questions, then that is it for today. So thank you.
Thank you.
Thank you.